So let's move on to part 2. And let's talk about these antibodies. Now that we've been far past the Ehrlich's experiment, where he found out they are proteins, but you can be a protein chemist, and you can work on the immune system, and you can work on antibodies, and that's what we're going to do, actually, where you're going to use them, but you're not going to have to worry about the antibodies, like taking over the lab, self-reproducing or anything, or being alive in any way. The antibodies are proteins, just like the protein that you eat in a protein shake or whatever. So, but what are they, and what do we know? And of course, we can use all the techniques we talked about in biochem, X-ray crystallography. NMR is a little tough because they're so big. NMR has a hard time seeing them. There's a lot of cryo-EM. In fact, for COVID, cryo-EM was the main technology, even more than X-ray crystallography, because it's faster. And so there are scientists. I want to point out this scientific illustrator, this is actually the cover of Cell Nature or Science, I forget which one. But so you see how the molecule is like lit up here and it's red over there and all the molecules are going over there. It's a wonderful scientific combination of art and science. So I just want to show you a few of her arts. This is something about actually replication. So you see the ribosome here, and it's going along, and this molecule blocks replication at this base. Wasn't this a great way to show that? You can show the replication as sort of a board game. You can even see, and this is, I said replication, and I always use the wrong word. This is actually, this looks like it's transcription. Oh, the viral replication. I was thinking viral replication and applying it to DNA level. Okay, but this means that this is actually the transcription level, but its replication can be applied at several different levels. That's what we're doing here. So transcription, this is a transcription inhibitor. And ribosomal frame shifting, that's how you really know what's going on. This molecule makes it click off, and you know a little bit about what all these molecules are now. I like it when they have enough accuracy that you can say, oh, I remember that from biochem. So here is a thing about describing oncogenes, driving malignant cell proliferation, and there are a number. Do you all know oncogenes, what that term means? What does an oncogene cause? Yeah, it's a cancer-causing cell. Cancer-causing mutation. So basically when cancer it's like you're playing out of tune, definitely out of tune with the rest of the body. And they're driving, the gene is like the fiddler who's making the other cells dance to his tune by driving their proliferation. And then this is from Chasing the SARS Variants. This reminds me of, I don't know if you've seen the Bugs Bunny cartoons with that, they had the big monster, I don't even know if it has a name, the big hairy red monster that looks like this. It reminds me of that. I think she's inspired by that. Or maybe Margaret the Martian, I'm not sure. But the whole thing is, red was the color of SARS very early on. If we could have assigned it any color we wanted to, but of course, dangerous, we associate that with red, so we chose red. But I want to say that was an artistic choice. They don't have color at that level, and it has no color on its own. And yet you use that to communicate. What I love about this is it shows the antibodies with their Y shape. So each of the antibodies, and notice that they are running, and you remember the constant region is down here. The variable region is the hands, and they're trying to grab onto the SARS by binding with their hands, and that is where the binding part of antibodies are. And here's one where he catches it, except this is another movie reference, You Shall Not Pass. So this one's meaning Gandalf, and this is the Balrog, right? But like Gandalf was able to at least stop the Balrog at great cost, this was able to stop the SARS-CoV-2 again. So these are all nice pictures, but we need to know the protein structures. We can handle beta sheets, loops. We can look at all these things. So we want to ask in detail, where are the atoms? Here are the atoms. Just remember the terms. An antibody binds an antigen, and antibodies are really big. This is actually just the one arm of the Y. So an antibody is actually 3 times as big as this. It has 2 arms like this, and then a constant region. This is the variable region. And so the variable region of the antibody dwarfs the antigen. It can bind to a much smaller antigen. And it forms a protein-protein interface. that fits together like a 3D puzzle. And not just shape matches, but also charges, including partial charges like polarities and hydrogen bonds. So we can take this, when I see this, I'm like, as a protein chemist, I want to know where the atoms are and what they are doing. So the overall structure of the antibody is you've always got to keep in mind there's actually 12 domains in a normal IgG antibody, the most prevalent one in your blood. And there are 2 binding sites on it because there's 2 arms, each of which has an identical set of residues. It's actually dimeric, if you look at this, with a blue heavy chain that has 4 domains and a red light chain that has 2 domains. Then 2 heavies bind up, 2 lights are bound to them. They're connected by a couple of disulfide bonds in the middle as well. So this means reducing agent will actually affect antibody structure potentially. And they also have a little bit of bound carbohydrate, which means you can't just make them in bacteria, at least not the intact, full antibody. There's no carbohydrate up here. So bacterial-made fragments of just the V region actually could work, depending on a lot of other factors. So just realize that these are the terms that we're going to move back and forth when we talk about antibodies. And here's the binding site way up here. This is where the real action is. Everything else doesn't change that much. But if you have differences in antibodies and you want to have differences in binding, well, this is where you're going to have the variation. And that's exactly where you see it. So there are 2 ways of talking about antibodies. You can talk about the actual chains of proteins, light and heavy chain, or you can talk about what changes versus what doesn't. And that's why you have the constant region that doesn't change and the variable regions that do change when you're looking from antibody clone to antibody clone. So one of the biochemical consequences of this is an antibody's got 2 hands. If it's grabbing onto something with 1 hand, and if it's a repeated pattern in some way, it could be a repeated pattern on 1 cell, or it could be 2 different cells with the same pattern. It can bind 2 things. And this is actually really important to how it works, because if you think about it, if you are bound to 2 things, like if you're grabbing on, if you're hanging off the edge of a cliff, okay, holding on is kind of important to your future. If you're hanging on with 2 hands, you're probably a lot more secure than if you're just hanging on with 1. So that, and the antibody is hanging on to the virus with 2 hands. If it's able to do that, that if 1 hand lets go, the other hand's still holding. And we can actually see this. I've measured this in my lab in a certain way with our vehicle. We've actually tried to see the different situations. And yeah, it has about a 100-fold difference in binding. It's a logarithmic scale, but still 100-fold. So these are the IgG antibodies, and usually is what people are talking about. However, there are lots of different branches of the immune system. IgGs are just the most prominent and easiest to get to. So there's a lot to study in the immune system when you go to other parts of the body. And in fact, if you think about SARS-CoV-2, have you heard about they have a nasal vaccine? One of the reasons for that is that an injected vaccine in your blood is a bit distant from the site of infection in your lungs. A nasal vaccine is right exactly where it infects. And there are even different branches of the immune system that deal with mucosal membranes. And there could be even different kinds of antibodies. It gets really complicated. We're, of course, going to focus on the generic IgG. I just realized IgG for generic. That could help you to remember it, I suppose. You see that this is just in serum. So like in the serum, you have a lot more of IgG than the other kinds. You do have a little bit of this IgA, which ends up being 2 Y structures stuck together. And then there's IgM, which actually has 5 Y structures stuck together. Technically, there are 20 chains, because remember, there's 4 chains per antibody. But you can still see that even for all the differences of all these, they have different constant regions maybe, but they have the same basic Y structure. And so you can still talk about the constant region. It's just going to be a different constant region, but it's going to look the same from this distance. There are places where, and cases where these other IGs come into play. And these also involve different kinds of immune cells. So for example, peanut allergies actually get into the blood and trigger an immune response. The peanut allergen is an antigen that mast cells, which hang out underneath the skin, and they use IgE. They listen to the signal, they bind through IgE. And then they are the ones who release white blood cells under the skin. So this is a whole skin-based method. And the whole thing is they show the symptoms. Mast cells are very different, but on the mast cell membrane, you have this, and you can see IgE. Well, that's clearly an antibody. So what we learn about antibody structure in general does apply to these other branches. So why are antibodies bivalent? Well, one of the reasons is that they can do things that, better, like what you see on this slide. So antibodies can actually be made to more than just like viruses and bacteria. It can be made to anything that you want to get rid of. And sometimes there are toxins that you want to get rid of. It could be just a bad molecule, not a whole bad protein. Or it could be like botulism toxin, where the toxin itself is a protein. Whatever it is, these toxins are not alive. They're smaller than cells. But antibodies can be developed against them. And when an antibody does that, if it binds a repeating pattern, it can actually bind multiple toxins. They can sort of gang up, and they can nab the toxins and take them to jail, or whatever it is. There's also, if there's bacteria around, there's cells called macrophages that will actually, the antibodies will cook the bacteria, but the antibody by itself usually won't, it can block an interaction, like it can block SARS-CoV-2 infection, binding the receptor. But if it's just a bacteria hanging out and it just gets coated with antibodies, that's not that bad for the bacteria yet. That's why the macrophage coming along is important. It binds the constant regions, pulls the bacterium in, and then dismembers it, basically. Bacteria doesn't really have members, but it does, I think it can be dismembered nonetheless. So bacteria, there's even a case where the antibodies can bind a bacterium, and then there's a system called complement, which is actually a non-cell system. It's actually a protein-based system, and it might be one of the oldest. It's not as effective as our adaptive cellular immunity, but it is completely biochemical. So it's kind of like, I think of it like, the traps that have been laid for it, and you spring the trap and the complement goes and it punches holes. Because what you can see, one of the things that happens is the complement can actually form pores without the assistance of a cell. Really pretty amazing. But also at the same time, then a cell can come along, find the antibodies, and help out with the whole thing. Once you get ingested, the macrophages are really good. Notice the phage word. We have the phage word, which is the eating word. They eat and they chop the stuff up. Okay? So here, I want you to find the different immune cells. There are two different kinds of IgEs here. So what color did David Goodsell pick? He picked green for a bacterium. And in fact, you can see the flagellum right here. You can see sugars on the cell surface. So what else do you see that you can assign a color to? Can you find what the color of antibodies are? Can you find if there are different kinds of antibodies? And also, can you find something that's not an antibody that's doing something for the immunity? So, shoot, what do you have here? What do you see? Anybody see what color the antibodies are? Yeah. So we have pink antibodies. Sort of magenta pink. So there we have. And what's this? Remember the IgM? That was a pentamer. There it is. So you have at least an IgG and an IgM. I don't see any IgA, but I'm sure that that's somewhere.
What is the green stuff in the middle?
This right here?
Yeah.
Well, the one thing I know, number one, he colored it green, and number two, so I think he's coding it as some kind of invader, and you see antibodies that appear to be binding to it. So I would say maybe it's the antibodies are binding to sugars here. I don't know if he's implying that this could be some sort of a little bleb or like a little, what do they call it? Not a vacuole, but if you're an exosome, could be like an exosome, Or it could be just he wanted to show the antibodies doing something else, so he just put an antigen there and didn't really know what it was. I believe that the purple things are not antigens because you don't see antibodies interacting with those. Anybody want to guess? So you see the pore right here, so you have a bit of a pore. I don't see another cell around, so I assume the pore might be complement, but you can also have pores be made by immune cells as well, if there's one around. And the other thing is this red thing right here. Any ideas what the red thing is? You actually might know its name better than me. I just have an idea what its category is. Any guesses as to what the red thing is? I think it's some kind of fibrinogen, plasminogen, some kind of matrix blood clotting protein, like what they have. Somebody who's into that would be able to probably say exactly which one it is. So it's a blood protein, basically. But the thing is, okay, remember that each B cell starts off with a regular genomic copy of your DNA, but then it somehow makes an encoded, a gene-encoded, differentiated antibody. What it's done is it's taken its genes and it's shuffled them like a deck of cards. If you look in the part of the cell that makes B cell receptors and those make antibodies, you look for the code, you actually see like 8 different segments that you could combine, and you only need 4 of them. This is just an example. I'm not sure what the numbers are. But you have 8 different sections, and you inherit these. You actually do inherit these from your parents. And what you do is 2 of them are picked from this strand, 2 of them from that strand at random. And so you combine this, and just from these 8 different possibilities, I don't know how many different possibilities you have, but there's like at least 100 possibilities. If you just have these 4 gene segments, and if you also count for it, I believe you get one from your father, you can get them from your father or your mother, depending on recombination and stuff. Basically, this is directed recombination that shuffles these segments and puts them in order. And so we can understand, we can look in your genes and we can see something of what your immune response will be like. And so the other thing is when those go together, they make the Y-shaped thing on the surface of the cell. There's actually 3 modules, and I think that they're shuffled. So there's more diversity than this, but this gives you the idea of what it looks like. And then there's additional ways to have diversity where they join together. It introduces mutations deliberately. And finally, it just has a process where it just randomly introduces little mutations to try to make it better. So there are several ways to introduce mutations, but it does start from shuffled genes. So if you look at an antibody domain, this is the variable region, the light chain V region, and you sequence from residues 0 to 100. And you try to see where are the residues the same and where are they different. You can see that there's these red parts where you have a lot more variability. You have a lot more different residues. So they call these the hypervariable regions within the V region. I think we scan down, and you plot out where those regions are, and there are these little red regions that are on the end they're exactly at the antigen binding site. So this says that the antibodies need to be different. They need to be different at the tips. And those tips are at these 3 loops, CDR1, 2, and 3. It turns out that of these, in fact, you can even see it here, CDR3, it is actually the most diverse. If you look at it, it has higher red bars than the other 2. So there are 3 loops per domain, but remember that 2 domains come together, so there's actually 6 loops per binding site. And some of them come from the heavy chain, and some of them come from the light chain. So you have 6 potentially different loops. This is where I want you, I'm going to show you some data, and this is where part 2 of this is going to end. So I want you to look at this. This is some data about antibody diversity. We're just now getting to the point where we can sequence people's genes, and we can talk about what their antibody diversity is. And of course, we can sample their antibodies, and we can see what they actually make. So remember that you have naive, unique antibodies, and taking the gene segments together and sort of saying, how many different antibodies, unique antibodies, can I make from my genes? The answer is 10 to the 12th. In your blood, you only have 5 billion B cells. So you can see there's a big difference here. And actually, again, if you look at the numbers of Delano and Wells, they actually were dealing with numbers about like this. Remember, they didn't randomize everything. You can't randomize everything. That won't cover the space entirely. It also won't be as efficient as being more targeted. So how do people, you have to choose which antibodies actually get made. How do people choose? And how do different people choose differently? So they examined just 10 human subjects, but they examined them in incredible depth. Almost 3 billion antibody sequences is what they collected. And they basically looked at the patterns within those. These are the segments that an antibody actually goes through. So this is what they actually sequenced. And they sequenced the cards that get shuffled. So and these are the variable regions. There's called the V, the D, and the J, and then the CH region. And they talk about these are the genes that make the diversity. If you sequence these genes, you know which cards are on the table for each person. But then you also need to see which things make it through the gauntlet to be made by the body. So here's their first figure. Here's the 10 different people. Each person gets a color. I don't know if they were able to pick their favorite color or not, but it makes for a very pretty figure. And so you can see that there are some genes. Each of these is an antibody gene. And so this is a particular sequence that is a sort of germline sequence that you can inherit. And if you look up and down, there are some that, like, everyone seems to have this one. No one seems to have this one. And here you have, like, no one seems to have this one except whoever this green person was. Although the other ones had a few copies. But the main thing is you see a lot of variation here in the number of genes that you could get. And each person is different. And the cool thing is they even measured how many IgMs versus IgGs. And you know how we said that IgG was, and actually according to this, the frequency must be that this is more frequent. I'm actually, but I just noticed there's a different frequency for IgG versus IgM. I think that this means more frequent because I know that IgG is, maybe IgM has more genes. Anyways, whatever it is, there's a difference in frequency, but there's almost as much variation within people, the 10 people, as to how much frequency they have, as there is between the two of them. You know, the width of this line is almost the same as the gap between the two ends here, okay? So basically there's a lot of variation. Each person is different. You do notice that, you know, the blue person who has a high IgG has a low IgM, or vice versa. So what they found basically is that there are some favorite clonotypes, basically favorite antibody gene basic structures that are shared among different people. And the reason why the cells pick them is not because these are good at binding antibodies, but because they are good at developing into B cells, and these develop more often into B cells. So there's an immunological reason rather than a biochemical reason. And there's very large diversity. You have an incredibly large pool of diversity. That's why you can be infected by something new, and you can actually do well. So I want you to look at their data. So please do take out the handout that I gave to you. And I want you to look at the bottom here. This is the main thing to focus on, actually. This is the first thing to focus on. These are the most prominently shared sequences, and this is CDR3, where you see the most variation. So I want to look at this as a biochemist, and I want to say, which amino acids dominate? When you look at these, you can see certain amino acids dominating. There's a lot of variation, but you see patterns. So list the amino acids that you see dominating overall in the shared logo here. Then I want you to look over to here, and I want you to look at the frequency graph and see how the colors match up. The colors should match up: polar, hydrophobic, basic, and acidic. And they say in the middle, you have a lot of green. On the sides, you can have red and blue. And actually way out here you can have, but that doesn't matter as much. Basically, green in the middle, not many black in the middle. You've got red and blue on the sides. I want you to look for examples to confirm that those rules are followed. And then I want you to look for things that break the rules. You know, I see a few already that break these rules. So number one is about what's most prominent. Number two is about who breaks the rules. And number three, I want you then to compare this shared to a synthetic and an unshared. The synthetic, by the way, is just a projection from the from the genes without any B cell development or any immunological stimulation. So you can compare it to synthetic and to unshared and see what differences, what amino acids do you find in the other ones that you don't see as often in the lower ones. Okay, so I want to give you like a few minutes to work on that. I will give you a few minutes, and then we will discuss.